I'm speaking to you from Rio de Janeiro, Brazil, where we have a hot summer now. And it's a great pleasure to talk to you, to be here with lots of friends and lots of collaborators. Thank you for the invitation. So I'll be talking to you about this from Fuca to Luca. Fuca is a concept that we created ourselves. And we created the concept of Fuca because we examined the idea of a Luca, the last universal common ancestor. That was a concept described by Carl Woese to explain how the bacteria, archaeobacteria, and eukaryotes evolved. It was supposed to be the last universal common ancestor between these three domains, the big domains of life. Okay, and lots of works they have tried to understand what should it be the Luca's genome, the genome of the Luca. Okay, and how do these works work? How they manage to find the genome of Luca? Okay, they look for what we call the shared genome. What proteins and genes are shared between bacteria, archaeobacteria, and eukaryotes? And all the genes that are conserved in the three domains of life, they were supposed to be in Luca as well. Okay, and Luca and all this. This means that the scars of this ancient organism, proto-organisms, can be seen now in the genomes of the nowadays organisms. Okay, so all the time people are trying to make comparative genomics with this top-down approach, just taking all the genes from all the genomes and going back in the time and seeing what should be shared between all these organisms. And they found out lots of papers that Luca's genome presents already a basic metabolism machinery. He was capable to make glycolysis, citric acid cycle, and many basic biochemical routes. And also, most importantly, the Luca was capable to make the basic information machinery. It presents the basic informational machinery, the machinery for replication, DNA replication, transcription, and translation. Okay. The most significant work in this top-down approach nowadays, according to me, was this paper that was published by William Martin's group in 2016 in Nature Microbiology, on which they found out the Luca was an anaerobic carbon dioxide fixing, dependent on hydrogen, and inhabited a geochemically active environment rich in hydrogen, carbonic gases, and iron. Okay, but what I aim to focus in this slide is the following. Imagine that Luca, this very first organism that brings all life, the explosion of life, it presented 355 gene families. Okay, can you imagine an organism that is an ancestral, a very basic organism having 355 gene families? This is very sophisticated organism. This is not a basic organism. This is a very sophisticated organism. So we understanding this idea of the Luca, we came back. We want to came back to see what were the picture, what was happening in the earth, so that an organism like this with 355 gene families could rise. Okay, so we go to the bottom-up approach. That's what we have been talking in this conference until this moment. Okay, so in the bottom-up approach, we will understand the probiotic chemistry, right? How the basic molecules necessary to make proteins and DNA and nucleic acids in general, they arose. Okay, then we come to the RNA World Paradigm. Okay, and we truly believe that RNA rose much earlier than the DNA. Okay, and therefore the DNA replication is not necessary for the origin of life. And also the transcription was not necessary because there was only RNA at the first. So translation was the key to understand for us and for many others the origin of life in Earth. Okay, so we follow the ideas of the Rna, that was the Nobel Prize, the laureate of the Nobel Prize that studies the ribosome. So we want to study translation, and we believe that the ribosome was the very first molecular machine that was necessary to the origin of life. Okay, regarding the prebiotic chemistry, we have been talking in this conference until now about prebiotic chemistry. We have this preprint. This paper is like now 700 days under review, but we just review all these ideas about where each molecule necessary for the origin of life could rise. The ribose could be coming from comets and asteroids. The RNA polymers should be found in glaciers. The amino acids in volcanoes in many other places. The phospholipids, they could be raised in geysers. So we review all these ideas, and we call the prebiotic chemical refugia. The refugia are some places that we don't know which were they. The professor that came before me, they talk about a little bit of it. But the question is, there was some places in Earth on which the origin of the nucleobases, the purine, pyrimidine, the phosphate groups, the amino acids, they were facilitated by the chemical environment, okay? This is, we call the refugia, the prebiotic refugia. This refugia allowed the basic molecules to rise spontaneously, okay? So, and then we review in this paper. But then what happens, what we need to actually take on account is that the nucleotides, they arose. And also the amino acids, they arose. Somehow, we don't know exactly why. I think it's very interesting to discuss about it, but it's a fact that they arose. Otherwise, we wouldn't be here to tell the story, right? So, and then we believe that the tRNA, the transfer RNA, there are lots of evidence about it as well, is sort of the simplest and the most stable form of that the nucleic acid can like fold in nature, okay? So we believe that, and we have lots of evidence as well, that the tRNA was the first sort of molecular fossil, okay? And then the tRNA, Savio, my collaborator, co-author, and he'll be present at a conference here. The last one is from Savio. He found out that he created all the proto-tRNAs. He made this top-down approach. He got lots of tRNAs of lots of organisms, and he made a model to produce the proto-tRNAs. Which were the first tRNAs that arose, okay? And then he concatenated those proto-tRNAs, and he blasted these proto-tRNAs against the BLAST database. And what does he found? Incredibly, he found out that a couple of those tRNAs, these proto-tRNAs, these concatenamers of proto-tRNAs, they were very similar to the PTC, the peptidyltransferase center. What's the peptidyltransferase center? Is the catalytic center of the ribosome, okay? Therefore, we have this hypothesis that it's actually very well built at this moment, on which the concatenamer of proto-tRNAs were got together. Here in this picture, you can see this colored picture. A proto-tRNA of proline get together with a proto-tRNA of tyrosine, and a proto-tRNA of phenylalanine, a proto-tRNA of glutamine, and a proto-tRNA of glycine. And they made up something very similar to the PTC, that's the catalytic center of the ribosome. What's that? So in a prebiotic time, proto-tRNAs got together and they were capable to synthesize peptides. Okay, what does the PTC does? What the ribosome does? It got amino acids together. Okay, so from the proto-tRNA, we came to the PTC. Is the proto-ribosome. Okay, so the ribosome was built by a concatenamer of tRNAs. Okay, and then but at the time at the RNA world, we did not consider that life was created. Actually, when something happens in Earth that we can call it life. Finally, for us, we created the chemical symbiosis theory that life emerged for the first time when nucleic acids and proteins got together. They start to bind together. So there's not the chicken and egg. There was no chicken and egg problem in the origin of life. Neither the amino acids nor the nucleic acid, they are more important one than the other. Okay, just when they got together at this time, when the PTC started to bind amino acids in the median, okay, these were naked RNAs that were capable to bind amino acids, and the amino acids, they were very more abundant than the nucleic acids, because we know that they are created much easier in the prebiotic environments. Okay. So we published this paper, The Theory of Chemical Symbiosis, a Mogulian View for the Emergence of Biological Systems. And we are saying that the origin of life started with this chemical symbiosis, when the nucleic acids started to produce the proto-PTC, and the proto-PTC started to catalyze peptides randomly, okay? At the first time, randomly, okay? Okay, so the proto-PTC is the grandmother of LUCA. It's the great-grandmother of LUCA. It's the great-great-great-grandmother of LUCA, okay? So the proto-PTC is key for the origin of life. And then when this for us, we theorize, we create this new concept about the FUCA. What's FUCA? Is the first universal common ancestor. That's exactly that moment the FUCA is born, when the proto-PTC started to bind amino acids together, okay? So, and at that time, this binding was sort of random. We cannot say that it was random because the abundance of amino acids was not random, okay? So it was quasi-random, depending on how much amino acids there were in the prebiotic pool, okay? And then we suggested that the LUCA maturated. What was the next time, the very important next thing that happens in this prebiotic median, in this prebiotic idea? What happened was that the genetic code was created. It's very complex to understand how the genetic code was created. I think Professor Marco José will talk about this. We have models to say how the genetic code has been created by these proto-tRNAs. We don't have the complete picture at this moment, but we know that sometime the genetic code has been created, because it was very important. So actually, the creation and the maturation of the genetic code is when life actually inaugurates. There are two moments. When the FUCA was born, that was in this very first moment when the PTC binded amino acid. The second moment when the genetic code has been established. Okay, I would need a complete lecture to say how this happened, but I will just go ahead. So this completed the chemical symbiotic process that was completely self-assembled and self-organized to create this first organism, proto-organism. It was not an organism, it was a naked sort of RNA that was capable to create now proteins in an organized fashion with codons and codons code-defined for amino acids, okay? So this was the origin of FUCA. And that shows the relevance of the organic codes. We have published a couple of papers about the organic codes as well. And the idea of an organic code comes from the biosemiotics. The biosemiotics would say that life is represented, is understood, can be described as an interplay of codes, okay? But the code biology is slightly different. It's not the interplay of codes that's actually relevant, but actually the origin of this code and the fact that this code is a process that characterizes life, okay? And life exists through coding processes that operates molecularly, operates in the molecules, okay? And what's a code? The code is something in which the coding rules are not dictated exclusively by the laws of physics and chemistry, okay? The code has something of randomicity, and there are arbitrary rules that were self-organized to allow the correspondence between the world of RNAs, the world of nucleotides, and the world of amino acids, okay? So we would need the presence of a translator or an adapter. That's the ribosome, okay? So, and then from FUCA, what happens after FUCA? FUCA evolved then from, so FUCA was this first organism capable to bind amino acids and capable to have a genetic code. From FUCA to LUCA, we have an entire gap that we don't know what happens from FUCA to LUCA. What happens was progenotes. Progenotes is a concept created by Carl Woese again, okay? They were open biological systems, and we are now creating models about how the progenotes evolved and how the FUCA could become LUCA, okay? So what we know at this moment, they were open biological systems. They are not encapsulated at first moment, okay? So they were free naked assemblies of ribonucleoproteins. And on the world of the progenotes, there was the independent evolution of biochemical pathways. Each sort of population, lots of populations of progenotes maturated and evolved given biochemical pathway, okay? And this happens by the linkage of these random proteins that were generated, and then they didn't become random. These proteins, these first proteins, they bind together again the ribosome, and they stabilize the ribosome, and they bind back also some molecules that were present in the prebiotic pool, okay? And then in this progenote era, we have the lateral transference of information. It was an open system so that they transfer information very easily. Okay, they accumulated biochemical pathways and enzymes. Natural selection was operating there. Okay, these RNAs, they would bring to the whole system some sort of stabilization, okay, avoiding the catastrophe of error, right? And that was the origin of metabolism. We are now writing a paper exactly on the origin of this metabolism and the formation of the first genomes, okay? So in the world of the progenotes, we need to build the biochemical pathways, okay? And there is two questions. Did those pathways evolve by invention, from de novo invention, from nothing to something, or they were guided somehow by the nature, the physical nature of molecules, okay? So we believe that the both things happened, but there was, we believe, and there's evidence about this again, that they were guided. And how the prebiotic molecules guided the evolution of the metabolism. They guided because there are evidence now about what we call the protobiotic pathways, that glycolysis, the citric acid cycle, the pentosic pathway, they happened without enzymes. There are lots of growing evidence that the metabolites, they were capable to bring to malate, to oxaloacetate, to other, it happens. There are papers describing that the glycolytic pathway, the citric acid pathway, they happened before enzymes were created, okay? And then what happens in our view? The molecules started, the proteins, the peptides started to bind those molecules and bind and transform these molecules as well. And as much they were binding and transformed, they would be selected by natural selection. Of course, there are lots of holes in all the story, but you know, this is the general model. We want to take a big picture and understand from the zoom out what happened. So we believe that the protobiotic pathways of molecules, they guided the evolution of ribozymes and enzymes, okay? And then the next step happened when biology became encapsulated, okay? So the molecules ceased to be naked. So they ate the fruit of the biological tree of good and evil, and they were ashamed to be naked, and they got encapsulated. How this happened? First, we need to understand that viruses, they do not need cells to replicate. In our view, viruses are much earlier, they arose much earlier than cells. Why? Because they are much simpler, okay? Capsids are order of magnitude simpler than proteolipidic membranes, okay? And viruses, they are ribonucleoproteic as well as the ribosome, okay? And to control lipids in this first moment is irrelevant. It was not necessary, okay, for us, okay? And then viruses as well, they do not need cells to replicate. They need ribosomes to replicate, okay? Cells need ribosomes, and the ribosomes, they were abundant in the prebiotic media that we are proposing to exist. Viruses are not parasites, okay? Viruses are alive because they manipulate in the present organic codes, and they have proteins encoded in their nucleic acids. Therefore, they're alive. So viruses, the proteins of the capsids, they evolved by the evolution of next slide. Okay, they evolved by lipid binding proteins, okay? At some point in the origin of life, that pool of molecules arose a protein capable to bind lipids, okay? And then this protein capable to bind lipids, they were evolving and being natural selected. And even nowadays, we know that the membranes, they are highly proteic, okay? So this is something that happens in the origin of life. And we also believe that there was no LUCA, because actually there were two LUCA, or at least LUCA was not cellular at first. Why we say that? Because when we see how the bacteria, they build membranes, and how the archaea, they build membranes, they are very different, okay? In this slide, you can see that the phospholipid from archaea is in a different carbon of the glycerol group than the bacterial, okay? And one has an ester linkage, the other has an ester linkage, and also the side chain of the archaea, the lipid has a side chain in archaea that there is no in bacteria. So we believe that LUCA was not cellular, and they got different pathways to become the bacterial LUCA and the archaeal LUCA, that they are the Lucella brother. And we renamed the LUCA by the name of Lucella because of the last universal cellular ancestor. It's not the common ancestor, it's the cellular ancestor, okay? And all these pathways, they were already built in a genome of LUCA. Okay. Okay. So the whole model we are proposing, I'll just would like to invite you to read our papers because it's very few time to explain everything that I should explain here. But the whole model, we start with self-replicating proto-tRNAs and free amino acids. Then we go to the chemical symbiosis, where this replicating proto-tRNAs got together to produce the proto-PTC that were capable to bind amino acids together, producing quasi-random peptides. Then we got to the emergence of the genetic code on which the nucleic acid, this is the origin of our life, actually, when the origin of organic codes. Okay, then we got to the progenodes, to the progenodes on which the quasi-random protein started to bind these metabolites that were present in the probiotic soup, okay, and to build the genomes, right? And then we, at some point, we've got some protein that is capable to fit together and produce this capsid, okay? So we have the origin of viruses. So the virus evolved a lot. And after that, we have LUCA, when all this basic machinery of cellular machinery were already together, capable to work somehow. And they were finally encapsulated by this lipids that were binding to proteins. Okay, how the lipids come to the story? They came to the story with proteins that bound to them. Okay, because the origin of life is ribonucleoprotein. There are no lipids in the origin of life. Okay, but there is later in the origin of LUCA. Okay, so that was the just short story. It's lots of story to just tell in 20 minutes. We published the book. It's in Portuguese. It's now in translation to Spanish. Okay, and also, as someone said before, I also published a book, Que es la Ciencia, in Spanish. It's a book about the origin, the science, the philosophy, and history of science. Okay, and then finally, I would like to thank so much my dear friend Savio Farias. It's his birthday today. Congratulations, Savio. I would like to thank as well Professor Marco Jose, on which I've been there 1.5 years in Mexico. I did a postdoc. I'm very grateful to Marco, to Miriam, that is a student of Marco. Miriam, thanks so much for this invitation. Thanks, Sohan. Finally, we met us. So I share some papers with Sohan, but we never met even by internet. Thanks, Sohan, for this invitation. I'd like to thank the Brazilian funding agencies, FAPESH and CNPq, that finance my research, and the copyright owners of the pictures present here. Okay, thank you so much. I'm now